Nasodren, also called sinew forte, is a lyophilized cyclamen extract used for the treatment of rhinosinusitis. The prevalence of chronic rhinosinusitis affects about 15% of the adult population. It is estimated that more than 100,000 million people in the world suffer from rhinosinusitis. After many years of research and development, Hartington selected Cyclamen europaeum from the Caucasus, preparing an extract from its tubers to be used in Nasodren. The first stage of the production process is to obtain tubers from the Cyclamen europaeum. To ensure the high quality of Nasodren, this stage is carried out following good agricultural and collection practices. By following these extremely strict practices, we can avoid any inconsistency of the product with respect to the plant extract. Once the aqueous extract is obtained, it is titrated in order to quantify the amount of saponins. This ensures all batches of Nasodren contain the same effective range of saponin quantity as previously defined through dose-finding trials. Thus, the titration process guarantees the standardization of Nasodren. Once the defined volume of cyclamen extract is dosified into glass vials, it is lyophilized to obtain the dry extract. The lyophilization process consists of the sublimation, drying of a product within a vacuum that has previously been frozen. The extract is frozen below its so-called eutectic temperature in order to avoid the formation of water coagula. Then, the pressure is lowered and enough heat is supplied for the water to sublimate. At this initial drying phase, 95% of the water is sublimated. Pressure is controlled through the application of a partial vacuum. The vacuum speeds up the sublimation process. In addition, a cold condenser chamber provides a surface for the water vapor to re-solidify on. Unfrozen water molecules are removed with a secondary drying process. At the end of the lyophilization process, the product has a residual water content of around 1% to 4%, and the material must be sealed to prevent any reabsorption of ambient moisture. Thanks to this lyophilization process, the cyclamen extract can be stored at room temperature without refrigeration or the addition of any preservative for at least 5 years. Preservation is possible because the greatly reduced water content inhibits the action of microorganisms and enzymes that would normally degrade the extract. Once the lyophilized extract has been obtained, it is controlled so that we can ensure it meets all predefined specifications. It is then labelled and packed together with a plastic vial of water for its reconstitution. Nasodren's highly technical manufacturing process is carried out in accordance with the Good Manufacturing Practice Guidelines, so that a Drug Master File number has been given by the FDA. The freeze-dried cyclamen extract, which looks like an amorphous cake, can be rehydrated with water much more quickly and easily due to microscopic pores produced by the process. This is especially important for pharmaceutical uses, as in the case of Nasodren. Since its launch in 2002, Nasodren has demonstrated its efficacy in the symptomatic treatment of rhinosinusitis through many clinical trials, whose results have been published in the most prestigious international rhinologic journals. Nowadays, thousands of people benefit from the efficacy of Nasodren, improving their quality of life.